one question that we asked is, can genomics inform this physiological complexity in process-based models? So can genomics be used to help parameterize these models? And this was sort of an obvious step for me because coming from Cornell plant breeding, where genomic selection is sort of king and takes up maybe 50% of our seminars there, I thought, oh, this could be a good idea. And these are the guys who worked with me on this project. And so it really served two purposes. The first was, okay, how do we parameterize models at scale? How do we overhaul the model to work for how we want it to with this short-lived plant? And so you can think of it as, how do we do this concurrently, and how does one feed into the other? How does the model architecture need to be modified to be able to take in genotype-specific parameters, and how do we parameterize these models? When we think about what is growth, this is sort of my theory, I don't know if it's right, but you can think of it as the genetics or the genotype sets up a blueprint, like a scaffold. And that's sort of set, right? But then the environment and how that genotype then interacts over time, that gives rise to this realized growth. And so this is sort of the theoretical framework that I went after this project with. Okay, so here's the overview. We do what modelers do, which is we first develop a model based on theory by going through the literature and seeing sort of what arises there. So I developed a developmental submodel that's based on the sequential emergence and development of leaves, based on thermal time. This has largely been published already. But then I'm having issues. How about this? Okay. So that's here. And then we want to know, okay, so we have this model, we have these parameters. Is it feasible to actually measure from empirical data? Because one of the tenets of biophysical process-based modeling is that we don't want parameters that don't exist in the real world. We want them to have something analogous that you can measure that is tangible. They have units. You can collect data and get at them. So they're not sort of fit from data. Anyway, so the second is, okay, so we can measure them. If we can measure them in the real world, can we also indirectly estimate them using genomic prediction? If yes, sort of modify your model then. So this is the part where we need to concurrently modify the architecture of the model to be able to best utilize these types of data. And then this is all under non-limiting conditions experimentally, and then we want to know how does it need to be modified to be able to modulate this growth model under mild stress. And for us, we focus mostly on water stress in this project, so everything is going to be really about water stress. And again, can we derive equations that describe this, update the model again, and then if we're happy with it, if it predicts well for us under non-limiting and water stress conditions, can we then use it to explore sort of in silico what's going on and try to understand how we can best utilize a model like this. Okay, so the main governing equation for this leaf submodel is the relative rate of growth. It's a pretty standard function. It's the log. So for us, we use the logarithmic derivative of a 3-parameter logistic growth function. So you can see this growth here, this growth curve. And then the 3 parameters are K, N, and R. This is, you can think of this as this upper limit, this asymptote. N is the initial value, and then R you can think of as a rate. And so in this model, each leaf gets what's theoretically a cell division phase and an expansion phase. You can think of it as the cell division, and they're overlapping in thermal time. So you can think of it as the cell division phases. So how does a leaf grow? It creates a sort of meristem, and it has a leaf primordium, and this is sort of like a pool of cells that has the potential to go and expand. And then the expansion part is really what you're able to measure if you're measuring area. So each of these phases are allowed to be modulated by the environment, and we think that they are modulated differentially. In total, there are 24 parameters to describe this leaf growth model, and it also accounts for resource allocation to the above and the below ground, because we have to hook it into the rest of the model, right? We can't just have this in isolation if we want it to work for sort of the whole plant and their interactions. And we make sure that each can be derived from observation. So, okay, how easy is it to do so? So we did a little pilot study with 3 morphotypes here, vegetable, turnip, cabbage, and oilseed. This is what these guys look like. So we came up with a little protocol to use a software called Easy Leaf Area, so we can derive these images and get information on leaf area from these non-destructive leaves, so we can continuously measure these leaves over time. And then we have to derive the parameters. So here I enlisted the help of my former office mate, John Cleven, who's a Bayesian statistician. And so we used a hierarchical Bayesian framework to be able to estimate these for each morphotype. And so what's interesting is that using a Bayesian framework allows you to get posterior distributions. So not only can you get a point estimate of these parameters, K, N, and R, per morphotype, you can actually get a distribution. So this, I thought, okay, well maybe we can describe uncertainty this way. Because one of the difficulties of working with, I think, for breeders and geneticists to work with process-based models is that we're used to working with distributions, because things have uncertainty. And process-based models, they are largely deterministic. So you get one play out through time. And when you're thinking about extending the utility of these models to populations, and you want to know, is one RAL better than another? Well, what does that question even mean? They're going to be different if they're deterministic, unless they're exactly the same, unless the line is exactly on top of the other line, right? And so what does it mean? Like, how different are they really? So in order to make probabilistic statements about differences, we need to be able to propagate uncertainty through the system. And so can we do this by estimating, by putting the uncertainty around the parameters themselves? So this gave rise to that idea, but we wanted to update the model with these parameters, and then we can see, like, okay, so we can simulate these after it's integrated to whole plant model. These are the individual leaves over time. They look good with the morphotypes under non-limiting conditions. R500, which is the oil seed, sort of stops making leaves because it's flowered before the end of the simulation. Okay, so again, we can measure these from observations, but whether we want to or not is a different story, because they can be very difficult to measure, and we're thinking about population of hundreds or thousands. This is not feasible, so we need alternatives, and one could be high throughput genomics, like some of the work that Mitch is doing. Another is maybe we can use genomic prediction to predict some of these. So this is really a method borrowed from the plant breeders and the animal breeders. It's a method that started in cattle breeding to increase genetic gain over time, in which you're taking a training set and you're estimating the breeding value, and you can apply that to a different set, basically a test set. So for this, I used publicly available data. There's a RAL population that's publicly available between R500 and IMB 211, which is this out of Wisconsin. Fast plant, it's been selected for really fast cycling, so it's not a crop that you eat. It's not a cultivar, because this thing flowers in like 12 days and it makes like no biomass. But it's a really great research tool because it makes the population sort of cycle through faster. And I used leaf morphometric data that was published by a collaborator a few years ago. So he measured leaf traits over time. So from here, we were able to estimate the posterior distributions for these parameters for all the 123 lines. And then we used genomic prediction on the median of the posteriors, as well as the rate and the shape curve, the rate and the shape parameters of the resulting gamma distribution. So it's kind of boring. I'll just move through this quickly. But really, the result was we did pretty good. So this is K, N, and R, the three parameters: median, rate, shape. So you can see for K, all three of these metrics work pretty well. And for N and R, it's really interesting. But if you estimate really the distribution, you get a better estimate and you get better accuracy than if you were just using the median. So this really supported incorporating uncertainty into these models. And so what we have then, again, the model updates, so we can have a deterministic simulation where every time the plant's like, oh, I've accrued enough thermal time, it's time to pop out a leaf, it goes and says, I have K, N, and R, these are my parameters, and I can do this each step, compute the relative rate of growth, and go on from there. Or you can incorporate the uncertainty, which are genotype specific now, and each time it's time to pop out a leaf, you say, okay, I'm not going to take the values, I'm going to sample from these distributions. So that's a way that we can incorporate uncertainty. And if you want to talk about more, we can, but moving on to environmental modulation. So again, one of the values of using process-based models is that you can make predictions under environments that you haven't evaluated under, theoretically. So in order to try to find the relationship between, some of these leaf growth processes and stress. We did we carried out an experiment under stress conditions, well-watered, and 2 different periods of drought for cabbage and R500. And the idea is to derive a relationship and then incorporate it into the model and test it. So we measured a whole whack of traits, and then we incorporated the relationship into the model, and then using the parameters that were derived from the non-limiting, the previous experiment, we asked how it did in a variety of traits. And this is soil water content, total leaf area, model conductance, photosynthesis, and transpiration. And we're pretty happy with how it performs. There's still work to be done in trying to get a more mechanistic understanding of it, because these traits are very hard to collect in Nebraska. So if you ever try to pressure bomb a brassica plant, it's horrible. Sometimes it'll like shoot out and hit you in the eye. So there was a little bit of noise in these data. But then we were able to, with this updated model, we were able to sort of explore it in the digital space. So here, this is an in silico experiment. This is not real life. What we did was basically kind of a sensitivity analysis in which we fix parameters that describe the other processes in the model, but we allow the parameters that are in the leaf part of the model to vary, but we allow that variation to reflect a real population. Okay, so that they carry sort of the correlations that you might find in a real population. We simulated under well-watered and water-limited conditions, and then we compared the results for the 40 individuals that didn't hit flowering time before the end of the simulation for different traits. And this is what the simulation looks like. So well-watered conditions, you get water throughout the simulation. Water-limited, there's a period in the middle where we're withholding water. And then these results here are showing the ratio of the values for that genotype under water-limited to well-watered. So some are very expected, so soil water content obviously decreases under water-limited conditions. Leaf area index, you get sort of a permanent decline even after recovery. They're still not, they still don't have the same value of LAI as in well-watered. But interestingly, so if you look at photosynthesis and the model conductance, so what the gray is showing you is the max and the min values for those 40 at that time step. And the individual lines are sort of the individual RILs that I've pulled out. And what you see is that they encompass the value of 1. And the value of 1 means that the value in well-watered is the same as water-limited. And when that group of genotypes encompasses 1, that means that some genotypes have a poorer value in water-limited and some have a higher, lower and higher. And same for stomodal conductance. And this, where it sits on that, in that space, really varies depending on which RIL you are. And this is not something that we put into the model in the beginning. We didn't say, okay, this RIL is going to do this. This is just how it played out over time. And so this is really an emergent property of the system. And so if you think about what this means, this differential response of a genotype to the same environmental conditions that we've imposed on it, this is G by E. And so in this framework, G by E is an emergent property of the system. So to quickly summarize, I'm going to run out of time. So you guys can read this faster than I can say it, but really what this means in the bigger picture is that it hopefully may provide a way forward to be able to use these process-based models at the scale that geneticists and breeders are trying to work with larger germplasm panels. Okay, so what's next? We want to apply this to the NAM population, the nested association mapping panel, which is under development. And the goal would be, of course, to predict performance of genotypes under untested scenarios. Very quickly, bringing trees, the model, into agricultural fields, so exploring G by E in cotton. This is a collaboration primarily between myself and Duke Paulai at University of Arizona. He and I knew each other back at Cornell when he did his postdoc with Mike Gore. And he is now an assistant professor. So the motivation behind this is pretty simple, and I think it's really well encapsulated in this title by François Tardieu in INRA in France. And he says that any trait or trait-related allele can confer drought tolerance, just design the right drought scenario. And it's a very provocative sort of sentence because it's true. Like, what does it mean when the drought is so complex, and this meaning of drought tolerance is not meaningful unless you have the environmental context. And so, okay, I don't need to tell this crowd about the complexities, but so really we want to know what is the role of process-based computer models, and how can this digital world inform the physical world, and how can the results of our experiments inform like the model updates that we make. And cotton is a nice study organism because it is actually a perennial tree. We just cultivate it as an annual. It evolved in the semi-arid regions of Mexico, so I didn't need to make a whole lot of changes to the model architecture, which made me very happy. This was also an opportunity to add value to an old data set. So Duke had published a lot of this data that I'll present during his postdoctoral work, but there were some sort of old data sets that were lying around on the computers that no one had worked with, and so we wanted to see what we could do with it and see how to apply the model with it. So what they had done was developed a high throughput. This was the early days of phenomics, so it's probably much better now, but this is a higher clearance tractor, and here are a couple of sensors, so they can collect canopy temperature and DBI plant height sort of indirectly on a large population at higher temporal resolution. And they did this on a recombinant inbred line population. Here's the study overview. This is our old schematic here, but here I show like sort of the different data